Hello, I'm Jack Rogers. I'm talking about new therapies for manganese associated neurodegenerative and cognitive leasioning for this webinar series on cognitive and behavioral neuroscience, kindly invited by Jack Nathan. So basically, I'm the director of neurochemistry at MGH, Mass General Hospital at Harvard, and so I'd like to talk a little bit about some manganese pathways for manganese neurotoxicity and some therapies that we have in offering, antioxidant from urate and our own drugs for abrogating the manganese pathways which generate a neurotoxic effect in neurons. Basically, I'll just introduce the concept of what manganese does when it's poisonous or neurotoxic to overexposed individuals. And we are trying to develop the pathway where we showed in our paper with Vivek, Venkat, and Rami in 2018 that manganese overexposure is interfering with ferritin and amyloid precursor protein translation to perturb iron homeostasis and can cause ferroptosis and neurodegeneration. So there are many pathways which manganese might produce toxic effect, but the one that we have studied is that it can interfere with the translation of ferritin, the iron storage protein, and the Alzheimer's amyloid precursor protein, which actually is an iron export efflux protein, as we discovered with Dr. Ashley Bush and colleagues, Dan Cosman. So what is very important to note is that manganese encephalopathy offers similar though unique pathologies to Parkinson's disease, while this gait disturbance has been clinically reported for over 150 years. Compulsive behaviors, running, fighting, singing, liver cirrhosis, symptoms of irritability, difficulty in walking, gait, speech disturbances, similarities between manganese neurotoxicity and chronic hepatic encephalopathy were first reported by Krieger et al. In 1995 in Lancet. So it's very important to try to work out how, by the pathway by which we can figure out that ferritin and amyloid precursor protein can detoxify from iron overload, as to how they might detoxify from manganese-induced iron overload, and then save neurons from this death, ferroptotic death. We show that manganese exposure disrupts APP translation in dopaminergic neuron model of SY5Y cells. Venkataramani, Vivek, 2018 Journal of Neurochemistry, from Göttingen, and who's collaborating with us. And then we have got the fact that sufficient APPS may no longer be available for its role to efflux excess manganese and iron as required to protect neurons from ferroptosis. So when you block translation of APP, you don't have enough APPS, and therefore you can't stop ferroptosis and cell death from that pathway. So we model that APP and ferritin, iron storage, iron efflux, translation activators, including urate, may relieve the burden of manganese overexposures to neurons, then causing clinical iron overload. Certainly in elderly people, translation APP blockers, APP mRNA translation blockers, remain a strategy to prevent amyloidosis in Alzheimer's. However, we're studying toxicities in people who are under 40 years of age, generally speaking, who are exposed to battery factories and manganese oxide and welding plants, children, working adults, not generally at risk for amyloidosis. So in the next slide, I'd like to kind of dedicate a lot of the memories of this work on iron transport and iron overload, which manganese interferes with, to Marianne Wessling-Resnick, a good friend, and Dr. Barry Poor, a good colleague, who at Harvard discovered pathways by which ferroportin can be the final step for moving iron out of enterocytes after dietary intake of iron onto the hepatic portal vein, whereas the iron is taken up through DMT1 on the villi on the gut side of the lumen. So these two colleagues did wonderful work showing the role of DMT1 and ferroportin in dietary iron movement and uptake. And in fact, Marianne did work with manganese also, which is very seminal. So ferritin and APP expression are protective, translationally controlled genes, both neuroprotective, and they stop oxidative stress, iron Fenton chemistry build up, and oxidative damage. So basically, they're regulated at the translational level by iron regulatory protein 1 and 2, which bind the 5' untranslated region of these two mRNAs for these two protective proteins. And you'll find that then iron comes in and can remove repression by iron regulatory protein 1s or poly-C protein and allow translation of ferritin then to store iron in surfeit in the cell. Iron efflux to APP. Now, what we showed was with Vivek that manganese and lead can actually interfere with this pathway of translation and therefore nullify or reduce the presence of these protective proteins in cells, therefore stimulate a kind of neurotoxic cascade. So here is the translation of APP and ferritin in a cytoprotective mode, preventing cell death of neurons when overexposed to excess manganese. Vivek and helped do this in the Journal of Neurochemistry paper. He was instrumental in getting that paper out, in fact. So what we have got then is a situation with iron regulatory proteins, poly-C binding proteins that can control the translation of these two neuroprotective proteins in the cell. And we showed very astutely with Michelineni, Sohan, it was a Sohan Michelineni, showed that the 5' UTR of APP in fact falls into an iron responsive element. And then it's regulated through this pathway of iron regulated translation, just like ferritin, actually. And there is iron regulatory protein 1, which is a cis-aconitase when it actually is in normal functional mode. However, IRP1, IRP2 control the translational repression in iron deficit of several iron genes, including ferritin and APP. Actually, APP and alpha-synuclein also in the recent work we did. So here is the central paradigm that APP is an iron export protein. And that was worked out by James Deuce with Ashley Bush and ourselves, that APPS can bind to ferroportin, move too much iron out of the cells. This is the kind of iron that you would get when manganese, for example, as I'll show you, can lead to too much iron overload. And that's how it exerts some toxicity to the cells. APP is an iron metalloprotein, copper zinc binding, and it's involved in iron export. Manganese and lead interfere with ferritin and APP translation to perturb iron homeostasis and cause ferroptosis and neurodegeneration. So this is the liver-based encephalopathy, which affects the brain, similar to Parkinson's gait disorder, although you can't cure it with L-DOPA like Parkinson's, and it's the globus pallidus rather than the substantia nigra. As for lead, we found that increased APP translation acutely protects neurons and saves them from heavy metal exposure. Manganese actually causes this encephalopathy, which is related to, but not identical to Parkinson's. It causes compulsive behaviors, symptoms of irritability, and psychiatric problems. Actually, I'll show you also a paper we published on IQ as well, but local exhaust ventilation systems are a source of manganese, as in battery factories and in welding plants. Iron is very nasty, and it's in excess for lungs. And iron oxide can expose and cause disturbances in lung function. We showed with Fudi Wang, his lab in Zhejiang University in China, but with me and my wife, Catherine Cahill, and Weiwei Liu showed that there is a environmental link between too much manganese, and there is actually a very useful biomarker in hair as a reliable indicator of manganese exposure. And we were able to prove that by meta-analysis, quite extensive, Weiwei proved that that IQ units are diminished when you have got exposure to manganese. Here you see that the meta-analysis, which was published in Environmental Health 2020, with Dr. Wang, ourselves, and Weiwei Liu was the first author, that you can show that there's a very strong correlation, a tenfold increase of drinking water manganese is associated with a drop in IQ, 2.5 units, while not taking in absolutely sort of taking this effect in stone. It's still indicative of events that might affect cognition with manganese. The actual pathway of manganese to cause cytotoxicity is through iron regulatory protein 1 control of APP translation, ferritin translation. And what happens is that manganese comes into the cell through its transporter and can, in the liver brain axis, prevent APP expression and no longer allow ferroportin to be facilitated for iron export. Thus, you have a big buildup of iron load in the inside the cell and cell death. Ferroptosis is the hallmark of this, as Vivek has done this picture very nicely. We actually showed that what happens experimentally is that you lose very rapidly from the cell the APP and ferritin, as you can see from this Western blot. At the same time, you diminish the cell viability or MTT values of the manganese. Associated cell toxicity occurs. And you get cell number reduced as well. You can see from this control panel that the APP is increased with iron, whereas it is decreased with manganese. So exact opposite. And I'll show you slides that point to manganese interfering with translation of APP and ferritin by the iron regulatory protein pathway. The same effect in rodents, rats and mice, that exposure to manganese caused limits to the APP and ferritin content, therefore generating conditions of cytotoxicity. And then the target indeed was the iron responsive elements in the 5' UTRs of ferritin and APP, as you can see from this experiment with luciferase and cat reporter genes. That both manganese in both ferritin and APP were reduced in the presence of manganese in terms of conferred gene expression. In the control vectors, nothing happened. But when they had the 5' UTR of APP, then you had inhibition of expression. This paper in Journal of Neurochemistry repeated another paper previously in Journal of Neurochemistry, wherein we showed that APP overexpression protects cells from manganese dose-responsive toxicity, as you see from panel D. And also at the same time, APP overexpression in APP 695 caused less oxidative stress to these cells. Same occurred for APP knockout fibroblasts, which no longer responded to iron-mediated rescue from manganese cytotoxicity. Here you see the effect of the manganese to kill the cells in the APP knockout condition, fibroblasts, whereas APP plus cells are rescuable by iron to protect from manganese toxicity, and also without iron they are less toxic also, as you can see from panel D. So overall, I want to point out that urate, which is a well-known antioxidant, might have some efficacy towards this process and protect cells from manganese. And this model shows that in presence of manganese, you target the 5' UTR of APP mRNA and inhibit its translation. It no longer can export iron, which builds up and causes a ferroptotic death. In urate, you offset these events. The model is that you might do it through the IRE of the APP, 5' UTR, IRP binding sequence, but that has not been tested. But I'll show you evidence that the urate actually does protect cells from manganese induced death. We work in Boston, close to the Zakin Bridge, which is here. If you're into nouveau architecture, and it's certainly better than what was there before with the central artery in Boston. But anyway, targeting urate in the PD Parkinson's disease can apply to manganese exposure. That's the question. So there's epidemiological evidence, neurobiological evidence, therapeutic development of inosine, which is a purine precursor of urate. And then also the question is, it's well known that the astrocytic NRF2 activation is protective in the antioxidant pathway, providing protection for Parkinson's. So we thought that maybe it might be useful for manganese poisoning or toxicity to nerve cells. You can see that in the pathway, actually the urate is a product of purine metabolism, and this shows a selective advantage that, in fact, urate oxidase mutations in primate evolution may have contributed to the increased presence of urate compared to the presence of urate in mice to provide added protection in humans. Advantage of higher urate, possible cognitive benefit, possible hypertensive benefit, and certainly definitely an antioxidant, as Bruce Ames in the 1981, he was very famous for his work on mutagenesis, but he also suggested urate was protective. The epidemiology of urate and PD has been tested. Unfortunately, it failed in clinical trials, but with Michael Schwarzschild, although it has got a lot of very elegant work from his lab backing what urate does and what it does to perhaps even in ALS cases. However, we showed that manganese, so inspired by this, Ningjia and Rashid Bakshi and collaborated with us to show that urate can actually prevent manganese from being toxic, as you can see in the graph here, and that the toxicity coincides with maintaining APP in the cytoplasm of the cells. So there's some indicative data now that urate can protect cells from manganese toxicity. This is a more careful study where MTT assay where we were able to show that manganese is toxic again, as you can see, from untreated to manganese. However, as you add urate by itself is somewhat protective. Whereas if you add urate dose responsively, it can restore some of the manganese loss by about 30% if you subtract from background. So it's very worthwhile us working on how urate may reestablish cognitive improvement in manganese exposed people. This is a summary slide showing that the urate may restore APP to reestablish cells from manganese toxicity. Finally, I'd like to introduce you to the concept of RNA-based therapeutics, where we have actually screened a RNA-based drug, which is a 5' UTR of H-ferritin, which appears to protect from the consequences of manganese toxicity. Antibiotics actually have been around a long time, and they have been in the front line of RNA-based therapeutics for many years. The large ribosome is a binding target of erythromycin in bacteria, for example. However, we have screened a drug, which is called BL1, from a library at the Broad Institute, that appears to protect the cells quite dramatically from manganese toxicity and lead toxicity by targeting the 5' UTR of ferritin, which is the iron storage protein of all mammalian systems. You can see that we screened the drug first by identifying that there was iron responsive element-like sequences in neurodegenerative transcripts like APP and prion. And when doing a high throughput screen to prion, we got some inhibitors, but then encounter screens showed that the inhibitors actually were activators of a analogous but different H-ferritin IRE. So it was a fortuitous finding. And so BL1 does inhibit prion, but it has the concomitant effect of activating ferritin. We screened it, and you can see these assays. It actually induced in this Western blot assay ferritin and provided protection in panel E, which shows a calcine cell viability assay, and also cell viability assays with MTT show protection by BL1. Finally, the BL1 was able to, had the capacity to restore manganese and lead from causing too much toxicity to SY5Y neuroblastoma cells. Medicinal chemistry optimized it such that the analog 498 was more potent to provide protection from manganese toxicity, as you can see in the graph here. So the future directions of our work basically are that APP is a metalloprotein, and other than being an amyloidogenic protein that causes A beta to accumulate in the brain as a biomarker for Alzheimer's disease, also you can take not just inhibitors to try to prevent amyloid, but you can take activators of the 5' UTR to increase APPS, neuroprotective APPS output, which appears to operate to protect neurons from manganese toxicity experimentally. So we recognize that many people have worked on manganese toxicity. Superoxide dismutase is a manganese-containing enzyme. There are many mitochondrial markers of manganese effects. So perhaps the restoration of APP and ferritin has knock-on effects to some of these other markers of manganese, but certainly we did find that BL1 and urate can provide efficacy, one BL1 through ferritin and urate through APP to restore cells from the trauma of manganese toxicity. And here is our team at the Medical Education Center at Longwood in Harvard. That's my wife, Catherine Cahill, myself and our students. That's Yonsi, Suresh, and Henry and Harry. And we are very grateful to them. And we'd like to really say that we'll take any questions, no problem, and enjoyed the opportunity of talking with you.